Hello, everyone. In our previous class, we discussed how our immune system may protect us from infectious diseases. Today, we are taking a different look at this. We will try to understand how pathogens may evade that immune response and then now produce the disease. The first slides, about the first 7 slides that you will see from now on, these are the slides summarizing key concepts, essential concepts, to better understand infectious diseases. And we're going to start with this one here. So when a pathogen replicates in or on the host is said to have an infection, right? The nature of an infection can vary widely with respect to severity, location, and type of organism involved in this process. You're already familiar with this concept here, colonization. I use that a lot when we're talking about normal microbiome, how microorganisms that live on our skin or the mucosal epithelium, they arrive to that place, they colonize that place, and they start to multiply there because they find the optimal conditions for them to grow. But these are members of the normal microbiome that generally will not cause disease, although some of them are considered opportunistic pathogens. That means a pathogen that takes advantage of a particular situation of weakness in the host to now produce disease. So for normal microbiome, we call colonization. But in the case of a pathogen, this process of these microorganisms arriving to our body and establishing itself by infecting a particular tissue, a particular organ, we're going to call that process infection then. Now, the infection sometimes could be subclinical. Subclinical means that the pathogen is not producing in that particular subject, it's not producing any symptom or maybe mild symptoms. It could be a symptomatic disease in which we can actually now appreciate not only symptoms but also signs of that infection. Now, what is the difference between symptoms and signs? Symptoms are subjective effects, the way that is described there. These are things that we cannot measure. We have to rely on the patient describing that. When a patient arrives and says, they have pain, they have nausea, they have a headache, those are things that the nurse or the doctor, they cannot prove that. They have to rely and trust the patient that they're saying the correct thing. Now, a sign of the infection is something that is objective, something for which the physician, the nurse has evidence of that, something that can be seen, like, for example, rash, pus production, swelling, fever, because you can measure the fever and check what is the body temperature. Now, I was saying that, it could be subclinical, it could be symptomatic. The same pathogen could be subclinical in some subjects and could be symptomatic in others. This process of infectious disease takes into consideration many variables that sometimes are associated to the pathogen, maybe, for example, the dose to which the person has been exposed, or also things related to the host. It could be a host that is able to produce a good immune response to control that infection, while another one maybe is not able to do so. So the same pathogen could have different, the infection by that pathogen could have different outcomes in different subjects based on all these multiple variables that may determine the final outcome. Now, the initial infection by a particular pathogen is what we call a primary infection. I'm going to explain this better through a particular example. For example, if you get the flu by the influenza virus. The influenza virus starts to replicate in the respiratory tract, It may start now to damage that respiratory tract, including the mucociliary escalator. If that now gets damaged, it's going to be easier for other pathogens or opportunistic pathogens to now arrive in the lower respiratory tract. And those that arrive later as a consequence of this primary infection that facilitates their arrival, those that arrive later and produce an infection after this primary infection, we're going to call those secondary infections. Like for example, a bacteria that may cause pneumonia after the flu virus starts to replicate in the respiratory tract. Now, this slide has additional concepts, important concepts that you need to understand. First, the concept of primary pathogen. This is a microbe or a virus that usually cause disease in healthy individuals. You have several examples there of primary pathogens. Yersinia pestis. This is actually not the name of the pathogen, but the disease that they're causing, right? The disease plagued by Yersinia pestis, malaria by Plasmodium falciparum, the measles caused by the measles virus, influenza that causes the flu, diphtheria caused by Corynebacterium diphtheriae, tetanus by Clostridium tetani, and tuberculosis by Mycobacterium tuberculosis. These are all examples of primary pathogens. On the other hand, an opportunistic pathogen is a pathogen that usually will do no harm to a person that is immunocompetent, whose immune system is working properly. Now, if by any chance the defenses of a person are now compromised, that person is immunocompromised. It could be maybe because there is an immunodeficiency, maybe because it's immunosuppressed. Now these opportunistic pathogens will take advantage of that opportunity to cause disease in that person that is immunocompromised. That's why they are called opportunistic pathogens. They are taking advantage of that particular situation. Some members of the normal microbiota are opportunistic pathogens. For example, Staphylococcus aureus, Staphylococcus epidermidis, some species of Pseudomonas, like I mentioned here, Cryptococcus neoformans, a fungi, that is also an opportunistic pathogen. And then now, let's go to these two concepts, virulence and later virulence factors. But first, virulence. Virulence is a concept that sometimes people confuse with pathogenicity, but these are two different things, what we're talking about. Pathogenicity is the ability to cause disease. Virulence, on the other hand, is the degree of pathogenicity. How bad is that disease? There could be different degrees depending on the pathogen that is causing that infection. And for example, we could be talking about, I was mentioning an opportunistic pathogen, Cryptococcus neoformans. There are different strains of Cryptococcus neoformans. The virulence factors will be the factors that this opportunistic pathogen brings with itself that could now aid in the process of causing disease. For example, it's able to produce a capsule, and you already know the function of the capsule. It can evade the immune system by preventing phagocytosis. Some of these strains may also bring, for example, the production of melanin. Melanin, in the case of this microorganism, has been suggested that protects this. This is a yeast, by the way. Protects that yeast against oxidation. Other potential virulence factors that this particular microorganism may bring is, for example, production of enzymes like proteinase, and then other factors also that could be considered virulence factors. So to understand virulence better, basically what we are saying here is that if you have a strain of Cryptococcus neoformans that brings maybe only one of them versus another one that brings many of these factors, the one that brings more virulence factors could be at the end more virulent, could produce a more severe disease. And that's what virulence is about. All of them, all the different strains that express different virulence factors, they all have the ability to cause disease. They're all potential pathogens, opportunistic pathogens in this case. Now, how bad is the disease that they cause? It may depend on the strain that brings more or less virulence factors. Here you have more concepts related to infectious diseases. Well, the concept of contagious disease. Contagious disease is a disease that can be spread from person to person. You will see how there are some infectious diseases that we will discuss during the semester in which, you know, the pathogen is not transmitted to another person. We will discuss several of them. Like, for example, tetanus. If a person contracts tetanus, Clostridium tetani, it's not going to be easy to transmit it to another person. So that would be an example of a disease that is not contagious. Another important factor in infectious diseases is the infectious dose. And I'm showing you that when we compare two different microorganisms, two bacteria that target the gastrointestinal tract. Shigella species can cause shigellosis, and then salmonellosis, a disease that is produced by Salmonella enterica. There are different serotypes that can cause that disease. In the case of Shigella, in order to, you know, in order for the disease to evolve, the person needs to be exposed to about 10 to 100 cells of this particular bacteria. However, in order for Salmonella to produce disease, it has to be, you have to be exposed to a larger number of those bacteria. You have to ingest about 1 million of them. The reason for that between these two particular bacteria is that this one will be more susceptible to the low pH in the stomach. So you have to consume a large amount of them so that maybe few of them can finally reach the small intestine and start the process of infection and produce the disease. More concepts that are important that you need to understand. The course of the infection disease. There are different steps, right? Different stages of that disease that the patient is going to go through. The first one is the incubation period. The incubation period is the time that pass between, you know, the infection with the pathogen and the onset of the first signs and symptoms. When those first signs and symptoms appear, then now we are in the illness phase. Now, sometimes some infectious diseases, this phase of illness could be preceded by something that we call the prodromal phase. Prodromal phase is a phase of very vague symptoms where the person is just feeling, it's not feeling comfortable. Like, for example, when we talk about malaise, that feeling of discomfort, that is a typical prodromal phase. Now, after that illness phase, the person may recover, convalescence, the process of recovering from that disease, or maybe the person could die as a result of that infection. Something interesting is the concept of carriers. Carriers are subjects that get infected with the pathogen, and then they develop an asymptomatic disease, no symptoms, no signs of the infection. But then now they can spread that infectious agent and they can transmit it to other people. Those are what we call carriers. And they could be infected with that pathogen for a long, long period of time. It could be as a result of what we call a persistent infection, a chronic infection, in which the subject, the pathogen is replicating in that subject, is not producing any sign, any symptom, but then now is shedding that pathogen somehow and is exposing others to that pathogen. Now, the infectious disease can be classified also based on the duration of the symptoms. It could be acute, and that means this is a very quick infection. The onset of the signs and symptoms of the infections, they develop very quickly. This is usually an infection that may last only for a short time. The person may recover, or the person may die. Strep throat, for example, is an acute infection. Flu, infection by the influenza virus, is an acute infection. Ebola is an acute infection. And now you have also the chronic and latent infections. These are both examples of what we call persistent infections. And what is the difference between the two of them? In a chronic infection, well, first, what is similar between the two of them? We're talking about infections here that can last for a long period of time in the subject. It could be for months, for years, or for the rest of their lives. A chronic infection, however, is an infection where the disease is going to be developed slowly, and then the pathogen is going to be slowly replicating in that person. However, when we talk about a latent infection, we're talking about an infection in which the pathogen may infect a particular tissue in the body, and then now it could remain dormant, silent in that tissue for the rest of the life of the subject. Sometimes, it could be reactivated, and as a result of that reactivation, then now the pathogen starts to replicate again and may produce signs and symptoms of the infection again. Typical examples of chronic infections, for example, hepatitis B virus, hepatitis C virus, they can produce a typical chronic infection. Latent infection, herpes simplex virus, varicella zoster virus, human papilloma virus, those can produce latent infections. We can also classify the infectious diseases based on the distribution of the pathogen in the host. We have the case of localized infections. That means the site where the pathogen starts the infection is the site where the infection is going to be limited. It will be limited to that particular area. For example, Staphylococcus aureus causing skin infections like a boil, or also human papilloma virus causing infection in the epithelium. Those are localized infections. Now, systemic infection means that the virus is gaining access to the bloodstream or the lymphatic system, and is now spreading through the body. This is probably one of the best examples. Measles or HIV can also be considered a systemic infection, or dengue virus produces a systemic infection. Now, when you find pathogens or products of the pathogen in blood, we're going to use this suffix, "-emia", to describe them. For example, if there is bacteria present in blood, we will call that bacteremia. If there is a toxin produced by a bacteria present in blood, we will call that toxemia. If a virus is circulating in blood, we will call that viremia. And then finally, I have this concept here that is related to, a systemic process. Sepsis, we're going to be talking about septic shock in the future. But sepsis basically is an acute, life-threatening inflammation that could be caused by different infectious agents or by products of those infectious agents that are circulating in the bloodstream. But it's basically severe inflammation process. Now, we are done with this part of going through some key concepts related to infectious diseases. Let's now go through, who was the first one who established this connection between microorganisms and infectious diseases. The first scientist that thought about this and actually established postulates that guide the process of finding the etiological agent, the etiology, the cause of the disease, was this scientist, Robert Koch. Robert Koch, basically, that study using a bacteria, creating a pure culture of that bacteria. You already know what we mean by pure culture. And then now, the first thing that he said was that we should be able to isolate that particular pathogen from every person in which the disease is being diagnosed. And then that pathogen should not be found in people who are healthy. When that pathogen is isolated, a pure culture is created. And now with that pure culture, we can infect laboratory animals where, signs of the infection could be reproduced. For example, in this case, where we're looking at, the pathogen killing the host. Now, from that host in which the disease happened, now you should be able to extract that pathogen again and to create again a pure culture of the same pathogen that you isolated at the very beginning. If you're able to follow all those steps and you're able to prove that in all people who have signs, symptoms of the disease, you were able to isolate that pathogen and you were able to reproduce the disease in laboratory animals, and then at the end, recover that same pathogen, right? At the very end. Then now you can establish a relationship between that disease and that particular pathogen. Now, the only thing with these pathogens is that sometimes, it's hard to establish this relationship. For example, you may find that sometimes the organisms cannot be grown in laboratory medium. For example, the agent that causes syphilis, Treponema pallidum, still we struggle to grow that bacteria in the laboratory. Now, today we know that bacteria is linked to syphilis because now we have developed other techniques, immunological techniques molecular biology techniques that allow us to establish a relationship between the people that have syphilis and the presence of this particular bacteria. The other thing is that, in the previous slide, I was mentioning that you have to isolate that from all the people that have the signs, the symptoms, but not from those who are healthy. Sometimes the pathogen may produce no symptoms, no signs of the infection at all. And this could be, for example, a carrier who has a subclinical or asymptomatic infection. Another problem that you have to keep in mind when you work with these postulates is some diseases are polymicrobial. They're not produced by one microorganism alone, but a group of different microorganisms. For example, like the bacteria that produces caries. It's not a single bacteria, but several of them working together. And then also, sometimes you don't have an animal host where you can replicate that disease. For example, HIV, it's hard to reproduce that disease in animals, although today, some very special models have been developed in mice, for example, where, some events can be reproduced. So when you have any of these issues, it's going to be hard to use the Koch postulates. And then now you need to use different techniques in order to establish that relationship between the disease and the pathogen. As I mentioned before, it could be through immunoassays. It could be through molecular biology techniques, where you look at the presence of antibodies against that pathogen in all the individuals that have the disease, or you look at the actual presence of the pathogen by looking at the genome of that pathogen in the samples from people that have the disease. And then in that way, you can now establish that relationship. Now, let's now start with, let's now to try to understand how microorganisms are able to establish infections. We're going to start first talking about bacteria, then we will talk about viruses, and at the end, we will discuss a little bit diseases, the process of establishing infection in some eukaryotic pathogens. The first thing that needs to happen when a pathogen arrives to the body and is trying to colonize a particular tissue, and remember, if this is a pathogen and it's going to colonize, we're going to call that actually in an infectious process, okay? The first thing that they need to do is they need to adhere. They need to adhere to that particular, those particular cells or tissue that the pathogen is trying to infect. And for that, the pathogen will bring what we call adhesins. Adhesins will be molecules that will attach to host cell receptors. Who are these adhesins? Well, it could be, for example, part of the fimbria or the pili. It could be components of the capsule that is on the surface of that bacteria. It could be, cell wall proteins that are on the surface of that bacteria that can interact with the cell receptor in the host. Now, that binding is going to be highly specific, and it's going to look for a particular receptor, and this will determine what we call tropism. Only those cells, those tissues in which that specific receptor is present, those will be the only ones that could be infected. Now, the other thing is the whole process of colonization. Now that the pathogen arrived to the site, it has to establish itself there. And it has to establish in an environment that is going to be hostile to that pathogen. It's not going to be easy. Remember that we have right there the innate immunity, the adaptive immunity. They're going to make it impossible for that pathogen to survive in our body. However, there will be pathogens that will bring with them different mechanisms in order to evade this innate and adaptive response. For example, some pathogens may grow producing biofilms. Biofilms is a compact, mass of microorganisms, on top of each other, so that now those that are on the surface, they protect those that are deep inside of that biofilm, and that is a way for them to survive. You may find that, for example, some of these pathogens, they will bring with them proteins that we call siderophores that are able to bind iron. Why? Well, if you recall when we were talking about innate immunity, part of our innate immunity are the proteins and enzymes that we produce, right, in that first line of defense. Together with the physical barrier of the epithelium, we also produce some antimicrobial molecules. We produce some proteins, some enzymes. And I mentioned back then, for example, lactoferrin, transferrin. These are proteins that still take away iron, free iron, from the medium, not making that iron available to potential pathogens. But then now the pathogens bring a similar protein with the high affinity for that iron in order to take it back and to be able to use it. You may find pathogens that bring strategies to avoid or evade humoral response, like for example, IgA, the immunoglobulin that is present in the mucosal epithelium. There are some pathogens that may have these strategies that I'm mentioning here that could prevent, the infection by that bacteria. And a particular pathogen that does all of these together very well is the bacteria that produces gonorrhea. I will be talking about that one soon. So I will go through all these mechanisms that I'm listing here. And then the other thing is that the pathogen that is arriving needs to be ready to compete with our normal microbiome and to tolerate toxins that could be present in that environment. Now, once the pathogen attached to the surface, start the process of establishing the infection, some of them may need to cross some of the physical barriers in our body in order to find their final target. So these pathogens, they will bring different strategies in order to achieve that. For example, some bacteria, they don't have any particular tool to do that. And the only way that they can penetrate the skin is if there is any injury in the skin, any place in which the skin has been compromised, any cut, any wound that now allows that microorganism to enter into our body. Others, especially those that choose, the process of entering through mucous membranes, they may have a very particular mechanism to do that. They may promote the direct uptake by cells, and this is something you're seeing right here. Look at this cell right here. This is a bacteria, and that bacteria is inducing this process of engulf. Is asking that endothelial cells to engulf them so that they can now get into that epithelial cell. And they will do this by a set of proteins that the bacteria produce that goes interact with that epithelial cell, and then now produce a rearrangement of the cytoskeleton of that epithelial cell, and this ruffling of membrane to finally uptake that bacteria into the cell. I mentioned to you when we were talking about adaptive immunity, and we were discussing primary versus secondary lymphoid tissues. I mentioned to you this mucosal associated lymphoid tissue that is present in the gastrointestinal tract. Here you have a representation of the epithelium that lines the small intestine. And this is a single epithelium, and scattered among the epithelial cells, there is a very specific specialized cell that is called the M cell. If you recall back then, I told you this is a cell that is constantly sampling the lumen of that small intestine, and is bringing whatever arrives there, is bringing that into the subepithelial compartment and allow macrophages to take it up. Remember, macrophages are phagocytes. Through the process of phagocytosis, they will take that bacteria inside. And some pathogens, like Shigella, they are able to survive inside of that macrophage. So in a way, for them, this is an advantage. Why? When Shigella arrives to the lumen, in the luminal side of that epithelium, in this surface, Shigella is not going to find the receptor that they need to attach to. The receptor is in the base, right here, in the base of those epithelial cells. So now the bacteria needs to find a strategy to reach that, the base of those epithelial cells. And basically what they use is the M cell. They say, okay, M cell, you want to take me under the epithelium? Please do so, because I need to reach that particular area. And then now the Shigella has a strategy to survive inside of the macrophage. It will replicate inside of the macrophage, will kill that macrophage, and then now the bacteria that is released from that macrophage can bind to the receptor in the base of those epithelial cells. The bacteria can now enter the epithelial cell and now replicates there and is transmitted from one cell to another. Let's now go into the process of phagocytosis and how different pathogens can prevent this process of phagocytosis. Remember that phagocytosis is one of the effector functions of the innate immunity. And remember that this phagocytosis can also be promoted by the complement, because when the C3b receptor, the C3b molecules are produced, when the complement is activated, that C3b now can opsonize the pathogen, and the phagocytes, they have on the surface receptors of that C3b molecule that allow that phagocyte to grab, to take that pathogen more easily. And now it's going to be taken inside of the cell, inside of this phagosome, and the phagosome will fuse with lysosomes. And normally, after those lysosomes are fused and they start to deliver these digestive enzymes and these reactive oxygen and nitrogen species, that will destroy the pathogen there, right? And that debris can exit the cell through a process of exocytosis. Also remember that in the activation of the complement, C5a and C3a are produced, and these are chemoattractant. The role is to attract phagocytes to the site of infection in order to clear that infection. Now, you will find pathogens that they bring with them different virulence factors that will evade this process of phagocytosis. Some pathogens, for example, they may bring enzymes like the C5a peptidase that will destroy the C5a. If that chemoattractant is not there anymore, the phagocytes are not receiving the signal that is telling them where they need to go. At the same time, there are pathogens that the constitution of that pathogen, the presence of a capsule, the production of proteins. All of these things may evade the process of attachment to the phagocyte and that process of engulfment. If they have these structures, these proteins, then now it's going to be harder for that phagocyte to grab that bacteria and to take it inside. There are pathogens that basically they will escape out of the phagosome before the phagosome fused to the lysosome. There are pathogens that stay in the phagosome, but they inhibit the fusion of the lysosomes to the phagosome. And then finally, incredible, but it is true, there are pathogens that are able to survive inside of a phagolysosome. Let's take a look at some of them. So in the case of pathogens that can prevent opsonization, and in that way, it's going to be harder to be for the phagocytosis to happen. This bacteria, Streptococcus pneumoniae, has a capsule, and that capsule will interfere with the opsonization because it will, prevent the binding of active C3b. M protein. This is a protein produced by a bacteria called Streptococcus pyogenes. This is what produces the strep throat. This M protein that is on the surface of this bacteria inactivates the C3b complement subunit. And then now, opsonization is not happening. It will be harder for the phagocytes to take that bacteria inside and destroy it. Some bacteria, they bring on the surface a molecule that is able to bind to the Fc region of the antibodies. You already know that the antibodies, in order for them to neutralize, to immobilize, to have all these functions that we described before, the variable region of the antibody will bind to the surface of that pathogen, let's say, the variable region, with the constant region, the Fc region, facing out. With this Fc facing out, for example, now a macrophage that has Fc receptor could interact with that, could take it and destroy that inside of the phagocyte. Some pathogens, they bring on the surface of the cell, they bring a molecule that works as an Fc receptor. And then now the antibodies, instead of binding in the correct orientation like this, with the Fc facing out, they will be captured. They will be captured by this Fc receptor on the surface of that bacteria. And now the variable region is what is facing out. That antibody in that way is not functional. And then now the bacteria will evade this humoral response. Now I mentioned that some bacteria may arrive into the phagocytes, and once inside of the phagocyte, they could, somehow survive inside of the phagocyte. Some of them, what they will do is they will escape out of the phagosome. So they can escape from that phagosome before the lysosomes fused and produce the phagolysosome. An example of a bacteria that can do that is Listeria monocytogenes. This is a bacteria, it's a pathogenic bacteria that causes the infection that we call listeriosis. Listeriosis. It can grow and reproduce inside the host cells, and is one of the most virulent foodborne pathogens. People could contract that through raw or the supposedly pasteurized milk, for example. That's a common way to acquire this infection. Another one here is Salmonella. Salmonella that produces a gastrointestinal tract infection. The way that this bacteria will survive inside of a phagocyte is by remaining inside of the phagosome, but preventing the fusion with the lysosome. And now they could reproduce inside of that phagocyte, and they could be released later from that phagocyte to continue with the process of infection. And then finally, you have here something that I find really amazing. A pathogen that is able to survive inside of a phagolysosome. I mean, I cannot think about an environment that could be so unfriendly like a phagolysosome, in which many enzymes are being produced, a lot of reactive oxygen and nitrogen species are being produced there, and still in that harsh environment, you find this pathogen, Coxiella burnetii, that produces the Q fever. You find that that pathogen is able to survive there. Coxiella is actually an obligate intracellular bacterial pathogen, and it causes this disease that we call the Q fever. Not only can survive inside of the phagolysosome, it can also survive to standard disinfectants, and is resistant to many other environmental changes, again, like the one present in the phagolysosome, for example. Can be acquired via inhalation, and with an extremely low infectious dose could still cause disease. And the disease occurs in two different stages. There is an acute stage that presents headaches, chills, respiratory symptoms. And then there is an insidious chronic stage that goes after that initial acute stage. Most of the infections with this bacteria, they will clear up spontaneously, and they can be easily treated with antibiotics, tetracycline, doxycycline, and combinations of different antibiotics. And there is also a vaccine, I believe, available against this bacteria. It could be used to prevent that disease. Now let's look at these very interesting bacteria. Here we're talking about Neisseria gonorrhoeae. This is the bacteria that produces the disease that we call gonorrhea, right? Usually a sexually transmitted infection. However, this is a bacteria that is able to infect, colonize and infect many different epithelium. There could be infection in the eye, in the throat, in the genital tract, the anal area. So this is a bacteria that is going to find those receptors that bacteria needs to attach to in different epithelial cells. And this is a bacteria that arrives with an incredible arsenal of virulence factors that allow this bacteria to evade the immune response. Let's take a look at some of them by looking at this picture that I'm showing you right here. Some important surface antigens in gonococci. I'm going to call this bacteria from now on gonococci is a diplococcus. There are two cells there together, arranged together. These gonococci there are some important surface antigens in the surface of the gonococci that are usually associated to the structure that we call the pili. Remember, pili or fimbria, that has different functions, like, for example, attaching to a surface. And that's exactly what this pili is going to do in this case, okay? The major antigen of the pili is a protein that we call pilin. And the genes that codify for pilin in this bacteria, they undergo extensive gene conversion in a way that the bacterium can produce up to 1 million antigenically distinct P-lin molecules. So that means that this P-lin that is represented here in these tiny dots can be changed constantly. And if antibodies were produced against the P-lins that are there on display now, and the bacteria all of a sudden changes them to a different version, then now the antibodies that were produced before are not going to work against the new version that's going to be there. And then now it's not going to neutralize the bacteria. The immune system will try to produce antibodies against the new P-lins that are there on the surface. But by the time those antibodies are produced, maybe new versions of those P-lin are being presented there on the surface of the P-lin. Another mechanism that can happen here with this particular bacteria is that the bacteria changes in the production of some enzymes that could lead to chemical alterations in the surface LPS to now render the bacteria that is more resistant to the antibodies that are being produced against that bacteria. Bacteria also can release, look here. Let's say this is an antigen on the surface of that bacteria. The bacteria has a mechanism to release those antigens. If by any chance an antibody is bound to that antigen, the bacteria could now release that portion of the surface of that bacteria. And now the antibody will be taken away from the surface of that bacteria. We call this this particular process is the bacteria is releasing surface antigens in membrane blebs. And then now the antibody that bound to that antigen on the surface of the bacteria is being taken away from the surface by releasing those blebs. And another important mechanism that this bacteria has is able to produce an enzyme, an IgA protease, that will destroy the IgA that our body produces to neutralize that bacteria. Remember that IgA is the most important antibody, mucosal antibody, in the surface of mucosal tissue. And I already mentioned to you that this is where this bacteria, infects a lot of different mucosal tissue. The eyes, the conjunctiva, in the mouth, in the genitals, anal, different mucosal tissues where IgA is important in order to neutralize this bacteria. But because the bacteria produce this enzyme that destroys the IgA, then now the bacteria can survive there. Now, this particular slide, we are now going to, we are done with the part of how bacteria evades the immune system. We are now going to talk about the different mechanisms or tools that bacteria may bring in order to damage the host. Some of these mechanisms could be through a direct process. Like, for example, if the bacteria produce toxins that are killing our cells, that's direct. Another one could be indirect. Maybe the bacteria induces an immune response that instead of helping us, that immune response could be deleterious for our body. And I'm also, giving you an example of how this damage that these pathogens induce, it may help the pathogen to exit that host and spread to other hosts. Like, for example, Vibrio cholera induces watery diarrhea. And in that diarrhea, you will find millions of these bacteria that could now reach another host. Or Bordetella pertussis. This is a bacteria that infects the respiratory tract and triggers a severe coughing. With that severe coughing, more of those pathogens can be released into the air, and they could find other hosts to infect. Now, let's now take a look at this direct effect, toxins. There are different types of toxins that bacteria can produce. The first one that I'm going to mention here are the exotoxins. They're called exotoxins, and you may think, oh, they're called like that because the bacteria secrete that to the outside. Not necessarily. Sometimes the toxins are inside of the cell, and once those cells are lysed, now the toxin is released. When we talk about exotoxins, we are talking about molecules that are proteins that have a very specific damaging effect. Many different bacteria can produce, many different exotoxins, depending on, how they act locally or systemically. You could classify them in these two groups. And also you could classify them based on the target, on the, what is the particular system or tissue that they aim. If we talk about neurotoxin, we're talking about toxins that damage the nervous system. We're talking about enterotoxins. These are toxins that damage the intestinal cells. If we're talking about cytotoxins, this is just a general group of toxins that are able to damage a variety of different cells. The term antitoxin refers to the serum, the antibodies that can be created against that toxin. And then now that antiserum could be used to treat, an infection in which this toxin is being produced. Also, toxins, they can be used to produce vaccines. If you take those toxins and now you inactivate the toxin, then you create what we call a toxoid. And now that inactivated toxin that is not able to do any harm could be used as a vaccine, inject into the subject starts to produce antibodies against the toxoid that will also react later with the real toxin if that real toxin arrives into our body and could neutralize the real toxin. Examples of vaccines that use this strategy, many. Clostridium tetani, the tetanus vaccine uses a toxoid that bacteria produces. Corynebacterium diphtheriae produces a toxin, and for that particular bacteria, we use the toxoid, the toxin that this bacteria produce, as a vaccine to now generate neutralizing antibodies against that toxin. Let's take a look now at different type of exotoxins. The first one that I'm introducing to you here is the AB type toxins, exotoxins. That is a subunit A and a subunit B. You have the two subunits arriving to the target cell in which this toxin can do harm. The subunit B is the part of the subunit that binds to a receptor on the surface of that cell. And this now triggers the process of endocytosis. That means engulfing that exotoxin into that endosome. And once inside of the endosome, the subunit A will be released. And this is the subunit that will cause harm in the body. For example, some of these AB toxins, these subunit A may target the ribosomes and then now prevent the synthesis of protein in those cells. Of course, if a cell is not able to synthesize proteins, it's going to die. That could be, for example, an effect that this type of exotoxins could have on the cells. Then this is another group of exotoxins, an interesting group, superantigens. A superantigen is this particular type of exotoxin that some bacteria produce, like for example, Staphylococcus aureus, you have it right there, or Streptococcus pyogenes. They both are able to produce this kind of superantigens. The superantigen has a very unique mechanism to produce a harm in our body. You already know from immunology, from the adaptive immunity class, that there are antigen presenting cells that in the context of MHC class 2, they may present a particular antigen to a T cell, in this case will be a CD4 positive T cell, right? That has a very specific T cell receptor that is able to recognize that particular antigen. If the wrong antigen is being presented and this T helper cell is not able to recognize that, then now this T helper cell is not going to produce the cytokines, the signals that are required in response to that antigen. Now, a super antigen is a molecule that some bacteria produce that have the ability to force that interaction between the MHC molecule and the T cell receptor, no matter if they are not specific to that particular antigen. That super antigen will force that interaction and now will force that T cell to start producing the cytokines. And you could imagine that it's going to do this with so many different MHC T cell receptor interactions that all of a sudden is going to produce a massive release of cytokines in the body. And this massive release of cytokines will have a toxic effect in our body. It can induce shock. And we will talk more about this shock when we get to some of the many infectious diseases that can induce this shock and that can kill the host. So basically, as you can see here in this particular picture, right? The super antigen binds to the MHC class 2 molecule there, outside of the peptide binding group, not here in the peptide binding group, but outside, and simultaneously bind to the T cell receptor. It doesn't matter if that T cell receptor corresponds to that antigen or not, that super antigen will force that interaction and will force now this T cell to produce those cytokines. And there will be so many of them doing that, so many clones producing these cytokines at the same time, that this will basically overwhelm our body. And now this massive, this cytokine storm, this massive amount of cytokine that is being released from these T cells will have, a negative impact in the host. Now there is another group of toxins that we call, or the toxin, not plural, but toxin, that we call endotoxin, that you're already familiar with, endotoxin. You already know that endotoxin is the lipid A that is present in the outer membrane of gram-negative bacteria only. If a gram-negative bacteria, all of a sudden is in the bloodstream, let's say, producing a bacteremia, and then now our immune system start to fight that bacteria, and in the process of killing that bacteria, the lipid A gets released out of the cell. This lipid A is able to activate innate and adaptive defenses that result again in a massive production of cytokines that could have a deleterious effect in our body. This lipid A is also a pyrogen. I didn't mention that here, but oh yes, right here, can trigger fever. It's a pyrogen, an exogenous pyrogen. And also, this lipid A is very heat stable, and autoclaving may not destroy it. So you will never use endotoxins as a possible vaccine because we don't have a good way to inactivate that lipid A. It's very stable at high temperature. It's hard to produce a non-harmful version of that lipid A, so you will never use it as a potential vaccine against any bacteria, any gram-negative bacteria. And now here you have a side-by-side comparison of exotoxins with endotoxins, starting by the bacteria that are able to produce them. Exotoxins can be produced by both gram-positive and gram-negative. Endotoxins, is only gram-negative because it's that lipid A that is only present in the outer membrane of gram-negative bacteria. The exotoxins, they could be synthesized in the cytoplasm or they could be secreted to the outside. The endotoxin is an integral part of the outer membrane of the gram-negative cell wall. Exotoxins, they are proteins. Endotoxins, they are lipopolysaccharides. You can produce a toxoid with exotoxins. That means an inactivated version of that toxin, and now you could use it as a vaccine. Sorry. You could use it as a vaccine in the case of exotoxins, but you don't never use lipid A endotoxin as a potential vaccine, because you cannot generate a toxoid from this lipid A component. In terms of heat stability, you can easily inactivate by heat exotoxins, not the endotoxins. Each exotoxin, depending on the exotoxin that we're talking about, they will have a distinct mode of action to produce toxicity to the cells. Here, the lipid A has a very clear pathway for the damage that is going to be induced. It's basically, you know, activating innate and adaptive responses that lead to a massive production of cytokines and to you know, dramatic drop blood pressure, disseminated intravascular coagulation, then less blood arriving to vital organs, and then the person could die as a result of that. This can also kill, of course, okay, the exotoxins, but with a different mode of action. And then toxicity, in my opinion, you know, they are both highly toxic. We're done with the bacteria now. Let's now talk about viruses and how viruses, you know, can produce the infection and how they evade the immune system in order to survive in the host. Well, the first thing that you already know is that the infection by viruses happens by that virus finding the specific receptor that is expressed in the specific target cells. You already know about HIV infecting CD4 positive cells like T helper cells, macrophages, dendritic cells, or for example, hepatitis A virus infecting hepatocytes, because that's where those receptors are. And as soon as they arrive to that particular tissue or cells, they will attach, they will enter, and they will start to replicate in those cells. Now, the virus start to replicate, but soon it's going to find several mechanisms that will try to block that replication of the virus. Several mechanisms associated to innate and adaptive immunity. And here, you know, there are some of those mechanisms that viruses can use. Viruses can alter their antigens. Well, actually, before I go to the process of, altering the antigens, let's talk about interferon. Some viruses, they may block the expression of host genes, including, you know, those genes that could be activated by the, those genes that codify for interferons, or they may block the antiviral proteins that are activated by interferon. And now here, antibodies and viruses. Viruses, many of them, they may just avoid antibodies by different mechanisms. The first one here is, you know, instead of the virus being released outside to now find another cell, and in that trajectory, the virus could be neutralized by an antibody. Instead of doing that, some viruses, a way to avoid that will be to induce, you know, fusion between cells, so that now the virus can directly move from the infected cell to the new cell that is being infected. In that way, they don't have to go out. And this is the case of syncytium that may mediate this process of infection. Now this one, that I was going to jump at, immediately at the very beginning, because I think it's probably one of the most important mechanisms by which viruses evade immune response. Viruses can alter their antigens, when that antigen changes, then now is no longer neutralized by the immune response that was produced against the previous version of that antigen. The antigens affected are usually surface glycoproteins on the surface of the virus that are recognized by antibodies, but then also T-cell epitopes, could be also a source of variation for those viruses. The principal mechanism of antigen variation are point mutations and reassortment of RNA genomes, especially in RNA viruses. RNA viruses like the flu, for example, where you have the antigenic drift and antigenic shift. And this is a process that is essential in the spread of influenza virus. Now, the ability to produce these point mutations, especially in RNA viruses, that has to do with the fact that RNA viruses, viral and the viral RNA polymerases that they produce, they lack proofreading ability. So they are prone to mutate a lot, to change a lot. And some of these mutations could be an advantage for the virus to evade the immune response. Other viruses, like dengue, for example, they may actually use non-neutralizing antibodies that are bound to those viruses as a way to get closer to target cells, like monocytes, because those monocytes, they have Fc receptors that bind those non-neutralizing antibodies, and now the virus has a chance to get closer to that host cell and to enter into that cell. We call that antibody dependent enhancement. That happens in some viruses. This is now another mechanism that I think is amazing that some viruses do. And we're talking here about herpes viruses mainly. Doing this, like herpes simplex virus or cytomegalovirus. When these viruses infect the cell that they target, they will downregulate the production of MHC class 1. Remember that that MHC class 1 is vital to present antigens and to warn the adaptive immunity that something is wrong with that cell. Well, those viruses, they downregulate the expression of those genes, and then now these molecules will not be expressed after the virus infect. However, a cell that lacks MHC class 1 on the surface is an abnormal cell that could be killed by natural killer cells. But that is not that easy with these viruses, because these viruses bring a gene that expresses a molecule that looks like MHC class 1. It's a fake MHC class 1 just to trick the immune system. So now if a natural killer walks by and see that fake MHC class 1, is going to think that that cell is normal, that nothing is wrong with that cell. In the meantime, the virus is replicating inside that cell. So a very interesting mechanism that this virus has used to evade the immune response. So we are towards the end of this particular lecture, and now we are talking about eukaryotic pathogens, and I'm starting with fungi, for example. Fungi that can produce infectious diseases. I'm mentioning some of them here. For example, something to keep in mind, most fungi are saprophytes, and and those that can cause disease are general opportunistic pathogens. Many of them cause superficial infections, like infections in the hair, skin, nails. We call those dermatophytes. And the reason why they target these structures is that these are fungi that produce a very unique enzyme, keratinase, that will digest the keratin that is in our hair, in our skin, in our nails, because it's able to use that as a source of nutrient. It's a nutrient for them. And this is an important virulent factor for those opportunistic pathogens. Candida, a member of our normal microbiome, is another example of opportunistic pathogen, can cause disease in people that are immunocompromised. It may also produce, disease in cases of dysbiosis. That happens, for example, when women take an antibiotic and that antibiotic affects the normal microbiome, especially in the vaginal tract. Then now that yeast may use that as an opportunity to replicate more and to produce an inflammatory process and produce what we call vaginal candidiasis. We will talk about that when we get to genital tract infections. And then there are some fungal pathogens that produce toxins. Toxins that, because they are produced by fungi, they are called mycotoxins. Aspergillus flavus is an example of a species of fungi that is able to produce a particular type of mycotoxin that we call aflatoxins. Aflatoxins are poisonous, actually carcinogens and mutagens that are produced by certain molds, including these Aspergillus flavus. They naturally grow in soil or decay in vegetation. And then when contaminated food is processed, aflatoxins can enter the general food supply. And then now they could be found in pet and human foods. And children can be particularly affected by this exposure to these aflatoxins. And it could be associated with delayed development, liver damage, and even liver cancer. And then we're going to finish today's class looking at some other eukaryotic pathogens, in particular protozoa. Okay. The first one that I'm mentioning to you here is Plasmodium species, especially Plasmodium falciparum, associated to malaria. After a successful invasion, the malaria parasite, Plasmodium falciparum, extensively remodels the cells that they're going to infect. They can infect hepatocytes, but they can also infect red blood cells. They're going to change the architecture of that red blood cell. And they're going to provide this property of this ability to adhere to endothelial cells. This adherence to endothelial cells that is promoted by the parasite that is infecting the red blood cells plays an important role to escape immune mechanism. Why? Those cells now that get that are infected, those red blood cells that are infected with the parasite, and they adhere to the endothelial cells, they are now they will not pass through the spleen, and because they cannot circulate into the spleen, they are not going to be eliminated there. At the same time, this parasite is infecting red blood cells, and you already know they don't produce MHC class 1. So there is no way there to warn the immune system, to warn the T cells and tell them, hey, there is an infection going on in here, you need to destroy this infected cell. That cannot happen there. And then now we have this other pathogen pathogen that cause a disease that we call African trypanosomiasis. The particular bacteria, sorry, not bacteria, the particular protozoa associated to this infection, one of the species that is associated to this, Trypanosoma brucei. I will talk more about this particular pathogen when we get to central nervous system infections. But what I can tell you now related to this particular class is that the way that this parasite evade the immune system is by changing the surface antigens constantly. The surface of the trypanosome is covered by a dense coat of what we call a variant surface glycoprotein, VSG. In the genome of this parasite, of this protozoa, you find approximately 1,500 different versions of that antigen. What that means is that when the parasite enters the body, it may have this version showing up. Antibodies against that version of the parasite are produced. But then by the time those antibodies are produced, the parasite may switch to a different version of that surface antigen, that now these antibodies that were produced against the previous version, they cannot bind to this anymore. And then now the immune system needs to produce antibodies against these. But by the time these antibodies are produced, the parasite will switch to a different version of that surface antigen. And basically, it will be constantly, fighting with that humoral response. I'm not allowing that humoral response to produce antibodies that can finally neutralize that parasite.